we were just focused on brain growth in general because like you saw, you can kind of predict the size that the fly is supposed to be. And when we knocked out this gene, it's called ORB2. It's a RNA binding protein. We actually were looking at the other version of it, which is just ORB, and it's expressed like in the germline. That's what my lab focused on. The female germline and oocytes, the ovaries, things like that, and RNAs there. ORB2 was more highly expressed in the brain, and then literally looking under the scope when you dissect your first brain, that's a ORB2 null. So when you don't have the gene, it was dramatically smaller. And so then we started looking up, okay, what human developmental diseases or disorders, that's the same phenotype. And so mainly all we're looking at is microcephaly. We're kind of looking not at the known exposure to things that can, like Zika virus, that can cause pregnant mothers to, expose themselves and then therefore their babies to the potential of microcephaly, but more so looking for the genetic answer. Um, and then also our protein ORB2, it is orthologous to a human protein that has been clinically noted to be like deleted, mutated in human microcephaly patients. So that's kind of our clinical relevance, our link right now. And so what I think would be a cool direction from what I found is to insert the human version of the same gene into the fly and see do the same things happen. And then that would let us know, okay,
Mm,
maybe that could be something we add to like the prenatal screening. Maybe that's something to look at once we do see any like inclination that a baby might have microcephaly while it's still in utero.